So today, for I guess the whole morning, we're going to be talking about flow cytometry data analysis. And the way that we have it organized is that we're going to spend the first hour in the lecture hall, and we're going to give you guys an overview about what are some of the challenges in handling and analyzing flow cytometry data, and give you an overview of a couple of different methods that have been developed to take more computational approaches to assist in the analysis of flow cytometry data. And then we'll have a coffee break, and we'll head over to the computer lab where we'll have a couple of hours to play around with some of these software methods, these algorithms, so that you get a chance to have some hands-on experience in working with the algorithms and with the data. And so Tim Mosmann and I are going to be tag teaming the presentation both in the lecture hall and in the computer room. And so I'm going to start out, and then I'll turn things over to Tim. So how many people in here have actually done flow cytometry in the lab? So I guess most, maybe about 3/4 of the people, so that's good. So you have some experience in what flow cytometry is and how the data is analyzed. But just so we're all on the same page, and for some people who maybe don't have as much experience as others, flow cytometry, it's also known as fluorescence activated cell sorting, but it's essentially a method where we stain cell populations with fluorescent reagents that bind to molecules in and on cells. So for example, we may take an antibody that's conjugated with fluorescein that specifically recognizes the CD4 protein on the surface of cells. So we can stain those cells with those antibodies, and then we measure the fluorescent properties of each cell using this instrument called a flow cytometer. So by using this technology, that provides for the direct and indirect measurement of individual cell characteristics, including things like cell size, membrane protein expression, the expression of secreted proteins. DNA content can be measured and used to assess the stages in the cell cycle that the cell is transiting through. And it can also be used to measure signal transduction activation using phosphorylation-specific antibodies. So it's quite a powerful tool to look at how cells are responding to different kinds of changes in their environment and the other kinds of characteristics of cells. So we use flow cytometry for a variety of different kinds of use cases. We can use it to understand the differences in cell populations between different specimens. And that can be used to study normal and abnormal cell activation, differentiation, and function. We can also use flow cytometry to isolate cells from mixtures of cells. And it's used diagnostically in the clinical laboratories as one of the primary diagnostic tools for leukemias, lymphomas, and myeloproliferative disorders. So it's used both in a research laboratory as well as in a diagnostic laboratory. So over the last decade or so, the technology has advanced pretty dramatically in terms of the instrumentation to run flow cytometry samples, as well as the availability of staining reagents. And so now, with the appropriate combinations of different kinds of... Actually, is there a pointer somewhere? Pointer? Well, I guess I'll use my mouse. So with these kind of modern instruments, you can combine multiple lasers that will excite the fluorochromes at different wavelengths of light. And then when those are combined with different bandpass filters to measure the emission light that's coming off of the fluorescent reagents, now with these kind of modern instruments, the BD-LSR2, we can actually measure up to 19 different parameters on every individual cell using the appropriate fluorochrome reagents. And so what's happening now is that the data that we're able to collect on these cells is reaching higher and higher dimensionality, because we're able to measure more and more of these markers using the fluorescent reagents. Now, the way that we've traditionally analyzed flow data has actually not been keeping pace with the advances in the instrumentation. So the traditional way that people analyze flow data is still using manual gating of cell populations in two-dimensional dot plots or contour plots. And so a typical kind of manual gating strategy might look something like this, where you start out by looking at cells in 2 dimensions. Then the user will manually draw these hexagonal or these polygons around certain regions of the 2-dimensional data space. Then you can iteratively take this, the cells that are present in this region of that space and look at 2 other dimensions, and then further partition the data based on those fluorescent markers, and continue to do that until you've explored all of the dimensional space. But there's a lot of problems with analyzing the data in this fashion. So first of all, it's very subjective where you draw the boundaries of these polygon regions. And this is especially challenging when you have overlapping populations like this. It's hard to know exactly where you might want to draw this boundary. So it's very subjective, and what happens is that you'll get different results from different people who are analyzing the same data, because they might choose to partition those populations slightly differently. And it ends up being really time consuming as you get higher and higher dimensions, because now you have to first start out by doing this partition, then you have to take each of these populations and analyze them in all the other dimensions that have been analyzed in the data set. And then in these 2 dimensions, when you further partition the populations into 2, again, you have to take each one of those into the additional dimensions and examine them. And in some cases, you have to go backwards and make sure that the partitions that you generated remain to be consistent in the early dimensions that you analyzed. So it ends up being very time consuming, especially as the number of dimensions are increasing and the number of samples that are being processed are increasing. The other thing is that these approaches are very sensitive to slight differences in fluorescence intensity between samples. And we see that there are slight differences in intensity when you analyze samples on different days. Even when you're using the same set of reagents, the staining on different days can be slightly different and give you slightly different intensities in the fluorescent pattern that you see. And this is also a big problem when you're comparing samples in a clinical study where you have biological variability between patients and between their samples that can give rise to staining intensity differences. So there are a lot of challenges in using manual gating to analyze kind of the state-of-the-art flow cytometry data. And this problem is only going to become worse. I don't know if people here are familiar with this new technology that has been developed in the last couple of years. It's called the Siteoff machine, and it actually combines flow cytometry with time-of-flight mass spectrometry. And so instead of using fluorochrome reagents, they use rare earth elements to conjugate to the antibodies. And then they use mass spectrometry to quantify the amount of these isotopes of rare earth elements that are present in a particular sample. And essentially what they do is they generate a stream of cells similar to what you do with flow cytometry, but then that stream of cells is passed in front of a high intensity heat source that basically nebulizes the cells into a cloud of ions. And then it passes that cloud of ions into the time-of-flight mass spectrometer, and then it can measure the amount of these rare earth elements. And because that's very precise measurement, now they're estimating that they'll be able to evaluate somewhere between 50 and 100 markers on individual cells, because they don't have to deal with the problem of spectral overlap of fluorochrome reagents. So now instead of dealing with 20-dimensional data, we have to anticipate being able to deal with 50, 60, 70-dimensional flow cytometry data with the data that's coming off of these kinds of instruments. So manual gating is just not going to scale to these kinds of technologies. So several groups over the last couple of years have tried to tackle this problem by developing improved approaches for analyzing flow cytometry data. And essentially what we want to be able to do is identify cell populations automatically in the data, objectively and quickly in this multi-dimensional data space. and if possible, to try to, as much as possible, eliminate the need for manual gating or supervised analysis. And then once we're able to do that on individual samples, we want to be able to quantitatively compare these identified populations across different samples that might be captured across different experiments. So this was kind of the challenge that we wanted to be able to address. So, when a lot of the groups decided to try to tackle this problem, we first considered, could we use kind of conventional clustering methods that had been developed in the computer science community to solve this problem? And so one of the methods that people use to cluster data is called Kmeans clustering. And so I'm going to show you an example of Kmeans clustering and why it's challenging to use that method for the analysis of flow data. So this is kind of a mock two-dimensional dot plot of what some flow data might look like. In this case, it's pretty clear that we have 4 populations here, and we would want to partition the events into the 4 populations. And so with K-means clustering, you start out by telling the algorithm how many populations you expect in your data space. So we would tell the algorithm that we are expecting 4 populations, or K equals 4. And then it starts out by randomly selecting 4 events in this data space. And so it might randomly select those 4 events. And then what it does, it essentially partitions the space around those 4 events. So it will cluster these events, whatever events are closest to this particular seed, it will cluster those together, will then cluster these together, et cetera. And essentially you're partitioning the space between these different seed centroids. So it might look something like that. So after you do that initial partitioning, then it throws out those initial seeds and it calculates the center of the distribution of the events in this region. And so in here, the center of the distribution of the events might be somewhere around here. And that's called a centroid. And so it then calculates these new centroids for those partitions. It throws away the partitions from the previous iteration, and it calculates new partitions. Then it throws away the centroids, calculates new centroids from this partition, and it does this iteratively until it arrives at a stable solution. And so ultimately you might end up here, and then you do this final clustering, and everything looks great, right? But there's 2 problems with K-means clustering. One is that you have to know how many populations you expect to begin with. So if I guess that there are only 3 populations here, and I initiated the algorithm with K equals 3, you go through this process, and you end up with a result that looks like this. And this doesn't match my intuition for how many populations are present. But because the algorithm is initiated with random selection of seed events, even if you guess the correct population, you can get trapped in local optimum solutions. So this would be an example of that. So there are 3 populations here. I guessed K equals 3, but because I seeded the algorithm with these randomly selected events, I get trapped in this local solution that's not a global solution to the problem. So, as we were kind of working on trying to solve this problem and considering the use of some of these traditional algorithms, we ran into these kinds of problems, which made us quickly realize that we were going to have to come up with alternative strategies. So I'm going to tell you about the method that our group developed that we call FlowCAP. And Tim is going to tell you about a method that his group has developed called Swift. And then there are some other methods that I'll just mention briefly that are all designed to try to solve this problem of automated analysis. So with FlowCAP, what we've done is well, actually, let me just kind of step you through what FlowCAP does. So this is going to be a 2-dimensional example. But FlowCAP actually functions in n-dimensional space. So however many parameters you have, FlowCAP is going to do what we're demonstrating here in 2 dimensions in that higher dimensional space. So we first start out by partitioning this with a hypergrid into hyperregions. So we would do something like this, where we've partitioned each of these dimensions into 10 bins. with this grid generating 100 of these hyperregions. Then what we do is we say, okay, which of these regions would I consider to be dense, a dense hyperregion? That is, that it has a certain number of events in it that exceed some density threshold. So if the density threshold in this case were 3, then you would label these regions as being dense, and then these other regions as being sparse or unpopulated. And so you would identify where the dense hyperregions are. So then what you can do is you can merge together the neighboring dense hyperregions, and you would end up with something that looks like this, where you distinguish between this group and that group. Okay, so now this has done 2 things for us. First of all, it has told us how many populations that we're seeing in this n-dimensional space, and it allows us to estimate the center of the distribution of the different dimensions for each population. And so then we can calculate a centroid for the center of each of these distributions and just do a single round of clustering to give us the solution that we're looking for. So again, what this ended up doing is telling us, first of all, how many populations we're seeing, taking a data-driven approach, and it has estimated the center of the distribution of each of those regions. So that has overcome some of these problems with Kmeans. So this is the core approach of the flock algorithm. But to get this to work really well with flow cytometry data, the core of the algorithm is kind of embedded in the middle of a processing pipeline, where we have to first start out by converting the binary FCS files into a data matrix. We do some cleansing of some of these boundary events to clean up the data. We may need to kind of collapse the data into lower dimensional space for various reasons. We do a normalization step so that when we're calculating distances, that each dimension has an equal weight in that distance metric, so we can accomplish that by doing normalization. We do the flock analysis, and then we come back and calculate statistics and things like that. So this is, I think, in addition to developing the core of the algorithm, really to get it to work well in real-life samples, we have to incorporate this into this kind of a data processing workflow. So I'm just going to show you one example of what the data looks like when you analyze flock. And this is an analysis we did in collaboration with a group here at University of Rochester, Nyaki Sands. And they've been looking at the response of B cells in peripheral blood to different kinds of vaccinations. And they're our B cell immunology lab, and so they focused this analysis on different kinds of markers that distinguish between B cell subsets. So this was 10 color, 12 parameter data. It was initially gated on CD19 positive cells, so those are B lineage cells from peripheral blood. And we analyzed 67,000 events on average in these samples. And so when we ran flock on this 12-dimensional space, we ended up identifying 17 different B cell populations in these samples completely automatically. And so in this case, each of the different populations is color-coded with a different color, and this is how they're distributed in the different two-dimensional dot plots. And based on our understanding of B cell biology, what we're seeing here is kind of the differentiation pattern of B cells that are starting out in this region here, where you have the naive B cells that have not experienced antigen. And as they're going through antigen exposure and reaction, they tend to move in this direction through this particular two-dimensional dot plot, and ultimately end up in this region here, where you have the plasmablasts and plasma cells that are secreting antibody in response to vaccination. So, yeah, sorry.
Just a quick question.
Sure.
So do you set the threshold for the density to find a rare population like that?
Yes, that's a good question. There are 2 parameters that have to be used to configure the flock algorithm. We have to decide how many partitions to make in each dimension. The example I showed, we made 10, but you could choose 20 or 30. And you have to decide what that density threshold is going to be. And those parameters will influence the results that you get. And when we're at the computer lab this afternoon, I'm going to show you some examples of what impact that has on the results that you get. That's a good question. Okay, so one of the tests that we did to convince ourselves that we're really identifying discrete populations using this analysis is the prediction is that every population should be distinguishable from every other population in at least one dimension. If they overlap in all dimensions, it's not clear that they're really distinct populations. So this is just one example of that. So we have this region down here where you have these gray events, and you have this, whatever color this is, purplish. And they overlap, well, it looks like they overlap completely in that particular two-dimensional dot plot. So up here, I've just kind of highlighted the gray population. And if you look over here in this two-dimensional dot plot, I'm going to next switch to the purple population. And what we found was that, this is an example where even though they overlap, pretty precisely or pretty consistently in these two dimensions, the purple population actually expresses lower levels of CD38. So we went through and compared all of the populations and convinced ourselves that we could find that they were different in at least one dimension for each of those examples. And that convinced ourselves that we're really identifying distinct populations using the algorithm. So then we took a look at the 17 different populations and tried to figure out what does that correspond to in terms of known cell types that have been reported in the literature that people were used to evaluating. And so what we found is that we could identify populations that looked like they were naive B cells, unswitched memory B cells, switched memory B cells, other kinds of memory B cells, plasmablasts, and plasma cells. So we could kind of match these cell populations to known cell types that have been reported in the literature. But in addition, we were subdividing them further based on these additional markers. They really are real distinct populations when you look at the distribution of the staining intensities. We don't know what the functional significance are between these different populations, but I think this is kind of hypothesis generating type of investigation. So now we've seen these new populations, and now it's a matter of going back into the laboratory and trying to understand their functional differences. But anyway, so a paper describing the FlowCAP method and the analysis that we did on these vaccine responses of B-cell subsets was published in Clinical Cytometry in 2010, and so that's a reference if you want to learn more about the details of the algorithm and how it was evaluated. So one of the things that we've been very interested in doing is taking algorithms and converting them into software that's relatively easy for the experimentalist to be able to use. And I've been involved in developing a resource called IMPORT, the Immunology Database and Analysis Portal. This is a free resource that was developed through funding by the National Institute of Allergy and Infectious Disease. There's a lot of data that's being collected in IMPORT for people to be able to access and reuse. And there are a whole suite of different analysis tools that have been implemented within this infrastructure. And so when we are in the computer lab later this morning, we're going to use IMPORT to do some FlowCAP analysis of some public data that's available from other NIAID-funded investigators. Or you can use your own data. You can upload your own data into the resource and use these different tools. And so we'll take a look in more detail about the IMPORT resource. And so in the last couple of minutes, I just want to mention that in addition to FlowCAP, there's probably a dozen other algorithms that have been developed to solve these same kind of problems of analyzing flow cytometry data. So Tim is going to tell you about one of the methods that they've developed, but there are a bunch of other methods that have been developed by other groups. And the question is, are all the methods equivalent? Are some methods better than others? Maybe some methods are good for identifying rare populations, but are not so good for, quantifying frequent populations. And so what we decided to do is the people who were involved in this community, we got together and decided to figure out a way of comparing the performance of these different algorithms. And so we came up with a consortium that we call FlowCAP for critical assessment of population identification methods. And so what we've been doing through FlowCAP is trying to come up with some objective methods to test the performance of these algorithms in kind of head-to-head kind of friendly competitions. So we start out by collecting some de-identified data sets. We distribute those to the algorithm development community. They do their analysis, return the results back to us, and then we compare those results against different kinds of gold standards. And so in the first competition, we used manual gating as the gold standard to compare the algorithms against. And so we're right now kind of in the process of pulling all that data together, and we've submitted it for publication. But I think FlowCAP is now going to provide a really nice means for us to assess the performance of these different algorithms and figure out which ones are going to work best in different circumstances. So be on the lookout for hopefully this paper gets published soon. So I'm going to stop there, and I just wanted to acknowledge a few people who've been involved in this work. I have a really terrific computer scientist who works in my group, Yu Chen, who was involved in developing the flock algorithm. We've had a lot of help in implementing that with software developers at Northrop Grumman. We've had really terrific collaborative relationships with several people at the University of Rochester, especially Nyaki Sanza's group that I showed you some data from. And then these are the members of the FlowCAP organizing committee, including Tim and myself, that are trying to put these competitions together. And if you have any questions, there's my email address. Feel free to contact me. And so with that, I'll turn it over to Tim. If there are any questions while we're transitioning. Yeah.
So with your algorithms, there we can interface with Flow or something so that if you want to override the results of the algorithm, you can manually.
Yeah, that's a good question. So we're in the process now of trying to generate outputs that can be uploaded into FlowJo so that you can use that as a starting point and maybe do some refinements in FlowJo or use their visualization tools. But I know Tim's, they use FlowJo very intimately in their analysis, but we're working on that right now. Thanks, Tim.
Okay, so I'm Tim Mosmann. I'll tell you about an alternative approach to some flow analysis. And it's great following Richard because not only did he give you a very nice introduction to flow cytometry and issues there, but also a lot of the problems. Yes, thank you. A lot of the problems that he described very nicely are the same problems that we had to address, but we've gone in some pretty different directions. So same problems, different solutions. And in my title, I think the one key word in there is that we're looking at rare populations. And we set out to design a general flow cytometry analysis program, but because we were very interested in rare populations, we tended to use those as our guide to whether the program is performing well or not. And without really intending to, we built a program that was very good for finding very, very tiny populations. So the collaborators I'm showing here, Iftikhar Naim, Gaurav Sharma, are on the mathematical algorithm side. Jonathan Rebhan, Subkash Dada, James Cavan also contributed. And I just wanted to emphasize Jonathan Rebhan up there because he's been doing a huge amount of work getting all the computers and the programs ready so that hopefully they will run smoothly when we get into the lab. So the manual analysis that Richard described, there's sort of an extra problem for going after the cytokine secreting populations that we're very interested in. They're very rare, And as you can see from some of these plots down here, when you get down to an activated set of T cells, the cytokine production tends to be in, they're not plus minus markers. They tend to kind of smear out a bit. They're sort of low, medium, high. It's hard to decide where exactly everything is. And so when you start to slice and dice to get all the subpopulations, you end up with a lot of cuts, as Richard was suggesting. And one of my early versions of this looks like this. This was an analysis of one sample, and it's a Boolean analysis of all the different combinations of cytokines that can be made by different cell populations. And I like this. I found this useful. And a lot of my friends were extremely rude about it when I would show those at seminars. And it's kind of difficult to get into. So we went into this project with Iftikhar and Gaurav, and they were interested in the sort of more mathematical side of the problem of clustering these very high dimensional data sets. And I won't go through much of this because I think Rich has already explained very clearly some of the needs for automated processing. And just to describe the strategy that Swift uses, if we start with initial populations, some of those populations may be fairly symmetrical, some of them may be skewed, they can overlap with each other, and they can have a very high dynamic range. So you may have populations of 50,000 cells that are fairly homogeneous, and there may be a population of 20 cells that you're also interested in. So there's a huge variation in population size. So the first step, Gaurav and Iftikhar designed this step using the expectation maximization algorithm. I'm not going to be able to explain the mathematics in any detail to you, but I can tell you the principles how it works. But it's a model-based algorithm like the Kmeans that Richard described, where you have to tell the program how many clusters you expect, and then it optimizes that number of clusters to your data. So that can be very computationally expensive using this expectation maximization algorithm, even though it does a good job. So what they designed was an iterative process where, first of all, a small sample of cells are taken, that small sample is clustered, and that defines some of the major clusters. Then they go back into the data and take a new sampling, but this time they use a lower probability of sampling from areas that have already been identified as clusters and a higher probability from areas that are more sparse. So in that way, they can march through all of the data without ever addressing the whole mess of data all at once. And for the very large data sets that we often like to use, that's the only way to really get at it. And the result of that is that Swift scales very well with cell number. That twice the cell number takes less than twice as long to process. Okay, so that first step gives us a set of clusters, and it gives you back what you asked for. If you said 100 clusters, it gives you back 100 clusters. And they're a little bit weighted towards large clusters, and the EM algorithm will go after the larger clusters first. So the small clusters may be buried within the larger ones, or you may have got the numbers wrong. So our second step, we then take every cluster from the first step, and then it's independently examined, and we simply ask, is it bimodal in any dimension or any combination of dimensions? So that would identify something that needs further processing. If it is bimodal, then it is split using the same algorithm, the EM algorithm, but now, of course, in a much smaller scale because you're dealing with just one cluster. And it's split up until the point where everything is unimodal. And we're using unimodal as you have to set a stopping point somewhere. Obviously, even a small shoulder might be interesting, but we decided that to give us a reasonably good intuitive sort of boundary, we'd say that unimodality was our criterion. So as long as even if it's a lumpy peak, as long as it's only got a single maximum, then it's acceptable as a single cluster. So the splitting process then will go through a variable amount of splitting depending on the data. If you've given a very tiny number of clusters initially, then splitting has to do a huge amount of work separating everything out. If you've initially chosen a very large number, then splitting doesn't have very much work to do. Then that results in a lot of clusters, and because the original algorithm may have been very generous in fitting multiple clusters to skewed distributions, we then have to go through a merging step as the third step. And now this step also compensates in the other direction, that if we've started off with way too many clusters and the program has faithfully made all those clusters for us, then those need to be condensed back down again. And so the merging proceeds on the same criterion. Again, we're using bimodality as our test. If fusing two clusters would create a bimodal cluster, then we don't do the merge. If it makes a unimodal cluster, then the merge is allowed. Okay. And I borrowed a couple of slides from Richard just to show a different solution to the same problem. So he pointed out that you could get trapped in a local optimum, and what could be a single cluster might come through as two in the analysis. But of course, the purple and the green cluster on the left would merge in our merging step because they'd be unimodal. And this trapping of two clusters, because there was a symmetrical point right between them, those would obviously be bimodal and would be split very easily in the splitting step. So I think we can take care of those two situations. And of course, if we have the wrong cluster number in the beginning, that's easily taken care of again in the splitting and merging steps. Okay, a couple of comments on the merging process itself. We went through a number of versions of this, and I think we finally got now a nice robust version. One of the problems with merging is that if you assess all possible pairs and ask can they merge, then if you find that A can merge with B and B can merge with C, you're tempted to merge A, B, and C all together. But the problem is then you have linking populations. So here the small populations I'm showing in the middle, those populations will link the two large ones because everybody would be allowed to merge with everybody else and as pairs. And if you then did that, you'd end up with bimodal populations again. So what we do instead, which is computationally a little more expensive, but I think Jonathan's now got that beaten down to a very quick process, we merge sequentially. So first of all of the pairwise distances for all the clusters are calculated using the Kullback-Leibler divergence, and that gives a measure of the degree of overlap of the clusters. And the closest cluster pair is then examined. If it can be merged, it is merged, and then all the numbers are recalculated, and we do everything again. If that one can't be merged because it would create bimodality, we look at the next closest pair of clusters. And so that process continues until no more merging can be done, and that's the final result. And that seems to be a very stable result. And just a word on the kind of output that we get. I'd like to distinguish between hard and soft clustering. So if you're used to gating in something like FlowJo, that's a hard separation of populations because you draw a line, and if cell is one side of the boundary, it belongs to one population. If it's the other side of the line, it belongs to the other population. So that would be on the left, the hard separation. However, what we do with Swift is we end up, the final result from Swift is a probability distribution saying how likely each cell is to belong to a particular cluster. And so what we then do is use that probability to assign cells to the clusters. So each cell is assigned to the cluster, not necessarily where it has the highest probability, but the chance of it being assigned to that cluster is proportional to that probability. So what that means is that the final shape of the populations is very reasonable, but the cells that are actually assigned in there are they're stochastic, but they're stochastic with this underlying probability of how likely they are to belong to that cluster. So if you assign these cells multiple times, the same cell won't necessarily go into the same cluster each time. So that means there is real uncertainty about which cells belong to which clusters. But I think that's true because if you look at these figures here, the cells in the middle region, without further information, there's no way of deciding which population each cell belongs to. So you do have that uncertainty in the data. And I think the soft clustering recognizes that. The advantage is we then generate populations that are probably closer to the true distribution of the population, whereas the hard clustering, the populations aren't really shaped that way with a sharp line between them. Okay. And the problem, of course, is that when you go to cell sorting, the current cell sorters, I don't think, can handle the idea of the soft stochastic clustering. Okay. I mentioned that the program was self-adjusting so that no matter what number of clusters you told it you wanted, it would correct you and come back with a more stable number of clusters. And this demonstrates that behavior. So we took the same sample, clustered it many times with different inputs, all the way from 5 input clusters up to 500. And the result is that in the open circles you see that at very low inputs the splitting works over time, and you go from, say, 5 clusters up to more than 100. then those clusters are merged back down again to about 60. If, on the other hand, you tell the program to find 400 clusters, it will do that. The splitting generates even more because some of those are judged to be worth splitting. But then again, the merging process brings that down to somewhere in the region of 60 or 70 clusters. So we've got quite a good self-normalizing behavior. So that means that the objectivity that you really like has been restored. You don't really have to know how many clusters to expect. The program will make up its own mind about a reasonable number. Another feature of this is that you'll notice that all of these are identical runs on the same sample, but they don't give the same numbers. And that is typical of analysis of the high-dimensional data sets. And I think there is true ambiguity in the data. And so here's an example of a population of cells. And you might look at that and say, well, that looks like a cluster. Or you might look at it a bit more and say, nah, that shape looks kind of suspicious. I think there's maybe an extra cluster there. And you could decide maybe the bottom blue one is a separate one, or maybe the red one, or maybe you decide there's actually 3 clusters there. But I think that if you gave this particular problem to a number of manual operators, you'd get a few different replies back about what was really going on. And the program does the same thing, that something this close would probably, it might separate or it might not. And if it did separate, it might separate in the different ways that I've shown there. So that's a problem, because what we'd really like to do is not just cluster a single sample, we'd like to compare samples. So if we have 2 clustering runs and they have different numbers of clusters on even the same sample, then it's very difficult to match up the clusters. And there's another problem that's even worse for comparing samples by independent clustering, and that is on the right. I've shown 2 samples, and this is the kind of thing we do a lot of. One is an unstimulated cell population. On the left is a stimulated cell population, and you'll notice there's a small set of black cells, about 50 of them, that have appeared as the result of the stimulation. Now, the background isn't 0 on the right. There's 3 cells there. But I don't think there's any clustering algorithm that will find those 3 cells. You know, if I drew them a little closer to the red, they'd completely vanish. So, on the other hand, on the left-hand side, it's very easy to identify that cluster. So again, the cluster matching problem is very difficult, because if a cluster's there, it isn't there. You can't ask how many cells are in your background sample. So to fix that, what we've done is merging files electronically. So we do a single clustering run with a joint sample, and then separate things out after the clustering. So that way you force everything into exactly the same clusters. And a very easy way to do that, which is conceptually exactly the same, is to make a cluster template. And in fact, this is the last step of Swift anyway. You make a template and then assign cells according to their probabilities. You can take any number of samples and carry on assigning them to that same template over and over again. It's a very fast process, and that means that now everybody's within the same set of clusters, so you can do a fair comparison between them. Okay. Let me run quickly through a couple of examples of reproducibility in the program. So we're very interested to see how consistent the results would be. And this is an example of peripheral blood mononuclear cells, so a fairly rich population, pretty diverse. And what we've done is made a template on one of them, and then we've assigned cells in several different samples to that template. So the first panel there, P1A versus P1B, is 2 different assignments of exactly the same sample. And the number of cells per cluster is not quite identical, but it's pretty close. It's not quite identical because it is a stochastic process, so you get some small variations. The next one, P2 versus P3. P1, P2, and P3 are replicates of the same sample. So they're biological replicas, 3 wells in a staining tray. So I should point out that these are very nice replicates, and for that we have to thank Jason Weaver, who's sitting there in green and white, because these are his samples, and we like these samples because these are a very reproducible set. They're very well-controlled, high-quality data. So the middle panel, it's not perfect, but it's still a pretty good correlation. The third panel, P2 versus X1, is interesting because that's now comparing 2 different people. So these are 2 different normal people. So they're both PBMC from normals, and you can see there's a clear correlation, but now you see much more diversity between the populations. So I think the the whole flow cytometry analysis is quite sensitive when you burrow down to this level of detail and this number of clusters. Interestingly, if you compare two replicates from another person, X1 versus X3, now the correlation is tight again. So it isn't just that these other samples are kind of fuzzy and they're not going into the clusters very well. They're simply fitting in a different way, and that also has its own reproducibility. Okay. And we have similar results if we look at the median fluorescence of these clusters. So for each cluster, we can define a median fluorescence in each channel. And if we look at that median, if we have two populations shown on the right here that are a little bit different, let's say one person has a little higher staining with one marker, let's say their cells are a little brighter for CD4 than the other person, then what will happen is that the cells will probably still be assigned to the same cluster, but there'll be a little offset within that cluster. So I've shown there that the red and the black cells are not quite in the same place. So by looking at that shift, the ratio of the intensities in each channel, we can get some idea again of similarity between samples. And once again, if we do that for the same sample, two different assignments, it's pretty close. This is now the ratio that's being plotted here of the medians. Biological replicates in the top middle panel are also pretty good. You can see at low cell numbers per cluster, when you're down in the sort of 10 to 20 range, things scatter around a bit. But still pretty good agreement. But when you shift to a different person, in the bottom row, in the outside ones on the bottom, then again you see quite a bit more variability. Okay, so something I wanted to introduce here and then go through more later on in the lab is how we visualize these. And as Richard mentioned, we use FlowJo extensively. Partly we didn't want to go to a lot of trouble to produce a beautiful graphical interface like Richard did. That's a lot of work. I admire it, but we wanted a shortcut, and the shortcut we liked so much that we stayed with it for a while. And that is basically to trick FlowJo into plotting clusters for us, even though it thinks it's plotting cells. And the way we do that is to assign every cell two sets of values, its original private values, where it's got a fluorescence intensity in every channel, and we also give it as separate columns the median values of its cluster. So that means that for one particular cluster with maybe 500 cells in it, all 500 cells will have identical values in each channel. So that means when you plot them in FlowJo, they all plot on top of each other. So what looks like a single point is in fact 500 cells, 1,000 cells, whatever. And you can see that here in the top row, we've got conventional cell gating, and the sample's about 1.5 million cells. In the bottom row are the comparable gates stepping all the way through the same way, and the same number of cells, but now they're focused on the cluster medians, so you only see a few hundred populations. So basically one dot is one cluster. And that has some very strong advantages for us. Because we tend to separate into a lot of different clusters, we need a post-processing method that's capable of exploring that kind of diversity. And when the clusters are focused into their medians, it basically pulls in a lot of outliers to a single point, and that makes gating much easier. So you can catch a large population by catching a very, very small area in the middle of it. And in practice, that means that the gating is far cleaner, and you get through to the end result. And you can see in the final pair of panels, this is equivalent gating all the way through, except cells on top, clusters on the bottom. And using the cluster gating, you end up with a lot of activated cells here. So these are cells making interferon gamma, tumor necrosis factor. And there aren't many negative cells because we've gated on activated CD69 positive cells. The top panel, with the equivalent gating, you can see that the positive cells, the interferon gamma and TNF producers, are pretty similar. You can go one for one, they're almost identical. But the background of cells that we just can't get rid of by conventional gating is still there. Now that we can extend that a lot further, let me skip a couple of things. If we then look at the individual clusters that are found by this method, then it gets much more interesting. This is now clustering a concatenate of 6 different samples. 3 of them were DMSO controls, and 3 of them were stimulating with influenza peptide antigens. And so what we're looking for is influenza-specific T cells that are making cytokines. And this looks a little sparse, there's not a lot of cells up there, but the sample, the concatenated sample is about 8 million cells. So this is gated down from a very large population. And what you can see is now that there are 6 clusters here from left to right. And the top row, we've separated out the 3 controls. And if you look at the first 4 populations, the first 4 clusters, there's almost no cells there. But if you look at the influenza-stimulated ones, now you can see that there are a lot of cells, and those first 4 clusters all seem to be making TNF and interferon gamma. So we've got a very nice signal-to-noise ratio. It's successfully pulled out these clusters. And remember, this clustering was done with the joint sample with everything mixed together. The 3 colors represent the 3 replicates, and they're pretty well evenly replicated. So you may wonder why we have 4 apparently very similar clusters making the same things, and they've come through the gating procedure as well. But if we then look at those same 6 clusters in the bottom 2 panels and look at 2 different cytokines, the top is TNF and interferon gamma, and the bottom is IL-2 and a chemokine, MIP-1 beta. And then you can see that there is much more diversity. So we have number 1 doesn't make either, number 2 makes moderate amounts of IL-2, number 3 makes IL-2 and MIP-1 beta, and so on. And if you examine in enough detail, you will find that all these clusters have some difference between them. And so again, like Richard's sort of concern that you want to examine things just to make sure the theory really does work. Okay. I'll skip that one. Just to say that there are a number of other rare population applications or not so rare populations. Stem cells, we can isolate very nice diversity within a bone marrow stem cell population. Some samples from Ben Frisch. Also malignant populations in leukemia. What we did here, by doing concatenations and sampling of various samples, we can construct consensus samples or idealized samples in a lot of different ways. So it's a very flexible way of producing a reference sample. And what we've done here is produce reference samples from multiple normal people. And on the left are 3 normal people, each plotted in terms of cells per cluster against the average of the 3 normals. And there's a reasonable correlation there. They're all reasonably similar to each other. However, on the right, these are now leukemic samples, and you can see a very large number of clusters, and some of them with many, many cells that are present in the leukemia samples on the y-axis, but not in the mean of the normal on the. And you notice the log scale there, it's actually a very sensitive measure of those cells. Okay, so been through that. Let me just show you the sensitivity. I told you that we were interested in rare populations, so at one point we decided we wanted to know how sensitive we really could push it to be. And so we made a sample that was deliberately very large and very sparse in some of the interesting cells. So we put 18 different files together, and we deliberately used weak responses and negative controls, and we were trying to keep down the positive cells to very low populations. And that was pretty successful. They were very rare. And so we clustered that, came up with more than 1,000 clusters. But amongst those, I pulled out 3 that were in the activated CD4 T cell category. And you can see that these are very small clusters. The total number of cells, 25, 20, 21. And they were still showing nice diversity. The first cluster making TNF and IL-2, second one making only TNF, and so on. So the program does work down to that level. That was a mammoth sample. 27 million took a few days to run. But it will run it, and it will find them. And this, in fact, is a sensitivity of better than 1 part in a million for detecting rare populations. So I think we really did get what we wanted in terms of that sensitivity. Okay, so just a few summary points. That Swift is very good not only for finding things in multiple dimensions, but also for finding very rare populations. And it's, for me at any rate, it was a little counterintuitive that by analyzing everything in multiple dimensions at the same time, you end up with a lot less populations than if you go through a manual slicing and dicing procedure, because you're always losing little bits that where your gates aren't quite right in 1 or 2 dimensions. And so the multidimensional clustering actually gives you a simpler result than you can do manually. Okay, and one thing that I think we certainly had to come to grips with through all of this is that the idea that you can decide what each cell is, we've had to sort of give up on, because there is ambiguity in the high-dimensional data. And so we're now thinking of it being more important to identify the shapes and positions of the clusters without being quite concerned in the individual cell level. And I think that's a bit of a shift for some people, but I think it's going to be necessary because it's very hard to visualize the results of this. So you're having to trust the program to some extent, and we're not going to be able to do unambiguous classification of every cell. Okay, so I think I'll stop there, and I think we can take some questions, and then I think we have coffee and we can move over the way. We are actually on time.